Good evening, everyone. My name is Mansi Tripathi. I am TA for the course Applied Environmental Microbiology. So this is week 2 of the doubt solving session or the sample problem solving session. This is from the NPTEL course, and I am PMRF research scholar from the Department of Civil Engineering. My division is Environmental Engineering, and I am from IIT Madras, Chennai, Tamil Nadu. So what we will do in these courses here is that we will solve multiple problems which are related to that particular week of the lectures. So we will solve all these problems, and this is how we will go on. And in case of doubt you guys are having in between, I'll help them solving that as well. So the course content of this course is not much. However, these two subtopics are really big, so it will take 1 or 2 weeks. So, as it is bioenergetics and metabolism, and the second is microbial diversity. So, I hope you well understood what is bioenergetics and how we will construct the phylogenetic tree and everything in microbial diversity and what kind of different microbes are there. So coming to question number one is, like, I will give you some couple of minutes to solve all these questions, so we can go on like that. So the correct sequence for electron acceptors in non-aqueous plume that is being degraded by microbes starting from its center would be, okay, the correct sequence of electron acceptors in non-aqueous plume. Okay, the plume is non-aqueous that is being degraded by microbes starting from its center. Like we have started the microbes have started the degradation from the center and they are going towards the end. So what can be the correct sequence? Okay, so his sequence is for electron acceptors. Please keep that in mind, and it is non-aqueous plume. So please keep that in your mind as well. Okay, it is starting from center, means just a hint that lesser oxygen will be there, and then it is going out. So please try to find out answer of this question so that we can move forward with the next question. So I will give you a couple of minutes to solve this. You can give your answer in the chat box. Okay, so the correct answer for this question is fermentation, sulfate reduction, nitrate reduction, iron reduction, and aerobic degradation. So as we are starting from the middle, then we are going out. So in the stage of fermentation, there is no oxygen. Okay. Then second it comes sulfate reduction. So sulfate is electron there. Then nitrate reduction, iron reduction, and aerobic. In aerobic degradation, complete it is filled with microbes, oxygen. Okay, so this is how it is done. Hmm. Okay, please read this properly so that we can move forward with the next question. Okay, moving towards to next question. The central dogma of biology states these are four different options. Life started first as RNA. DNA encodes the genetic information which is transcribed into mRNA and then translated into proteins. Ribosomes recognizes an open reading frame by first recognizing the start codon. Bacteria undergoes binary fission. So which of the following that central dogma states? Okay. Please try to find out answer of this question. This is pretty simple question. I know you know the answer. Just give it a try and you can answer this question very neatly. The answer for this question is DNA encodes the genetic information which is transcribed into mRNA and then translated into proteins. So please take some time to absorb this question so that we can move forward to the next one. The next question is, consider the following half reaction. The reactions are of lactate and nitrate. Is it feasible for microbes to derive energy from using lactate as electron donor and nitrate and is electron acceptor? So these are the options. Please try to find out answer of this question. Okay, so please try to find out answer of this question. We'll see how it is solved in the very next slide. So please take some time to absorb this question. As we can see, the delta G naught value is negative in one case and positive in another. And however, you have to find out uh, the equations which all are written here are in the forward direction. So whenever these such equations are written, it's always there in the forward direction. So the delta G naught value which is given here, that too represents the equations in the forward direction. So please keep that in mind and solve this. So we'll see the solution for this question. So the answer is yes. G naught is equal to minus 104.49 kilojoule per electron equivalent. Okay. So please match your answer with this question and try to find out what you have done right or wrong to get the following answer, whatever you have got. So this is the next question. For any genetic element that are open reading frames, okay, so 1, 3, 6, 9. What can be the answer? This question is not very correct, clearly grammatically correct, but you know the answer. Okay, so this is very simple question. Please type the answer in the chat box. So the answer is number 6. So this is very clear solution. So we'll move forward to the next question. Which one of the following statement is not true about protein? So be very mindful, it is not true about protein. The primary structure of proteins is merely chain of amino acids connected by peptide bonds. Secondary structure of proteins may either be alpha helic or beta strands. Tertiary structure of protein is its 3D conformation and is sensitive to chemistry and temperature. Quaternary structures are the biological units of multiple protein domains but have no biological relevance. So, which of one of the following statement is not true? Okay, so the answer for this question is quaternary structures are the biological units of multiple protein domains but have no biological relevance. So, okay, so other three parts, that the primary structure of protein is what? It's a merely chain of amino acid connected by peptide bonds. However, when we talk about the secondary structure, it can be alpha helix or beta strand. Tertiary structure is what? Like when we say primary structure, it's like a 1D structure. Okay, different amino acids are there which are connected by peptide bonds. When we say secondary structure, so it's like a 2D structure. Okay, different kinds of peptide bonds, amino acid connected by peptide bonds will be there. It means like a 1D structure we are keeping next to each other when they are connected with alpha helix or beta strands. Okay, so when we are keeping it like that, so it is kind of xy in a xy plane. So it is a 2D structure, a plane. Okay, so tertiary structure is what? When different such planes are present in a Z frame, a what do we call it? A 3D conformation. Okay, so that is tertiary structure. And what is written? The quaternary structures are the biological units of multiple protein domains, means multiple tertiary structure, but have no biological relevance. So that is wrong. Obviously, they have biological relevance. So moving forward to next question is, a student streaked 100 unidentified environmental bacteria cells on a plate, which had grown exponentially to 1 lakh bacterial cells in 2 hours. Okay, what is the growth rate constant of these bacteria? Okay, so how do we solve these questions? First, you can write the equations, then we can move forward and solve these questions. This is a very simple question. Take some time to write all the values. Like we have n, we have n naught. We know n is equal to n naught e raised to power minus e raised to power k and t. We have time also and everything. So very simple question. I'll also solve with you. Please get me your answer. Okay, so how to solve this question? I'll use the tools for you. Sorry. So for approaching such questions, these are the numerical type of questions. So for solving these questions, pretty simple. We know that the bacteria grows exponentially. Okay, so n is equal to n naught, that is the initial value of bacteria, Okay. E raised to power kt. But we know that the bacteria undergoes binary fission, right? Okay, bacteria undergoes binary fission. So instead of E, there will be some other value, right? So, hmm, so it undergoes binary fission. So putting value of N as, so this will be the 1 lakh bacteria will be the value of N. And what is the initial bacterial count? 100. So 100 will be what? N naught. That is initial concentration of bacteria. What will be the T? Time is 2 hours. So this will be T. You can simply find out answer of this question. I have given one formula, but keep in mind it is for the binary fission. Okay. So take some time and solve this question. Okay. So shall we check? So the answer is coming as 4.6 per hour. But I know you guys might not be getting this answer because when we will put all these values, so here we are putting as 1 lakh. Sorry for the bad handwriting. Divided by N naught, that is 100, is equal to e raised to power kt. Okay. Then it will come out as whatever it will come out, then the k value will be something 3.4. So that will be the correct answer. So, so no deduction for this particular question. Okay. So moving forward, in logistic growth, dn by dt is equal to rn 1 minus kn. The bacterial population will stabilize at, what is the position in which the bacterial population will stabilize? Please solve this question. So the solution is 1 by k. Please match this question with your solution that we can move forward to the next one. We'll move forward to the next one. Which of the following statement is not true? Exponential stationary lag death. This is the phase of the bacteria growth. The second is net ATP generated in complete utilization of glucose during aerobic respiration is 24. So net means because we know in the process of glycolysis, some ATPs are consumed in the first phase, right? So net ATP means gross minus the consumed one. The correct sequence of bacterial population growth is lag, exponential, stationary death, which is like the first one, which is it is also a bacterial population growth curve is exobeta 1,4 glucanase catalyzed degradation of beta 1,4 glycosidic bonds in the outer cellobiose unit of cellulose. Please try to find out answer of this question. So the solution is net AP generated in complete utilization of glucose during aerobic respiration is 24. This is the wrong statement. So in the comment box, you can let me know What is the net ATP generated in complete utilization of glucose during aerobic respiration? What is the number of ATPs generated? So you can answer for them in the comment. We'll move forward to the next one. This is the match the following type question. Please try to find the answer of this question. Okay, the solution for this question is one will go to fourth, that chemoorganotrophy uses organic chemicals as source of energy. Chemolithotrophy will use inorganic, litho means inorganic chemicals as source of energy. Phototrophy, photo means light, so it uses light as primary source of energy. And autotrophy is like the plants are autotrophic, right? Phototrophy and autotrophy both are governed by the plants. So in autotrophy, what it fixes its own carbon from inorganic sources, like similar what plant does. Inorganic is carbon dioxide is an inorganic source, and plant consumes carbon from inorganic source, that is carbon dioxide. So please note it down so that we can move forward to the next one. The next question is most energy rich compounds primarily in cell are phosphorylated compounds with ester or anhydride bonds having G naught values less than the Gibbs free energy, standard Gibbs free energy values less than 30 kilojoule under exothermic reactions or pass through cellular membranes easily. Please try to find out answer of this question. Okay, the solution for this question is the most energy rich compounds primarily in cell are phosphorylated compounds with ester and anhydride bonds. Okay, please note it down so that we'll move forward to the next one. Moving towards next question. Basically, curved cycle is termed as aerobic phase of respiration because aerobic condition is essential for the continued operations of the electron transport system, that is ATS system. It consumes oxygen. Oxygen act as catalyst or none of these. So, curved cycle is termed as aerobic phase of respiration because it's pretty straightforward question. So the answer for this question is aerobic conditions are essential for the continued operation of the electron transport system, that is ETS system. Moving forward to the next one. Which of the following doesn't occur until the Krebs cycle? Acetylization, dehydrogenation, oxidative phosphorylation, or decarboxylation? So the answer to this question is oxidative phosphorylation. Okay, so it doesn't occur until the Krebs cycle. Okay, it occurs after the when the Krebs cycle kicks in. Other process starts when before the beginning of the Krebs cycle. Okay, so the next question is what is pyruvic acid in respiration? A result of protein breakdown, or broken down into two carbon fragments and carbon dioxide. One of the product is Krebs cycle, formed only when the oxygen is available. So, what is the pyruvic acid in respiration? What can be the answer of this question? Please let me know in the chat box. Okay, if you don't want to speak, you can just simply type in the chat box. We'll see how many people will answer, and we'll see what can be the correct answer. It's like an audience poll. So you can just mark in the poll also. Four options are there. We are talking about pyruvic acid and respiration. Okay. I'll give you a couple of minutes to solve this. Okay, we'll see the solution now. So the solution is broken down into two carbon fragments and carbon dioxide. I can see only two people have answered the question correctly. Okay, so no problem. So pyruvic acid is the broken down, it's a substance that is broken down into two carbon fragments and CO2. Okay, so this happens in respiration process. Please note this down so that we can move forward to the next one. So the question number 14 is, which of the following is true about glycolysis? It occurs in the mitochondrial matrix of microorganisms, or it occurs only in aerobes. Third option is, it is said to have two phases. The fourth one is, it does not consume energy. So we have discussed something like this in the previous questions. Remember, when we're talking about the ATP produced by the consumption of glucose. Okay, so please take that question as reference, as well as the lectures that you have seen in the past week. Please answer this question. You can use the chat box. So this is to remind you again that we'll be uploading all the lecture materials as well as this video that which is under preparation on YouTube, and the lecture material will be available in the form of Google Drive to all of you. So if in between session, if you have any question to ask, then you can write me. You can just text me in mansi.in. Okay. So I have already mentioned the website in the previous session as well as the beginning of this session. In every session, I'll provide my website. So please let me know if you have any doubts in between sessions. Eventually, we'll meet every Tuesday. But in case you have any kind of doubt, you can just text me. Okay. Please solve this question. So the answer to this question is, it is said to have two phases. Okay, so this is the true thing about glycolysis. And we know that it doesn't occur only in aerobes, and it does consume energy. Okay, it consumes some ATP and converts that to ADP, and in the subsequent process, we get more ATPs. Okay, so it does consume some ATPs. It's not energy free process. So we'll move forward. Which of the following pathways involves carbohydrate metabolism? EMP, HMP, EED, and all of the above. So while solving these questions, I want all of you to write the full forms of EMP, HMP, and EDs in the chat box. So solve this question and you get 3 more questions in this by writing the full form. So I just want this session to be one question connected to the next one like this, so that you can understand and revise all the concepts accordingly. Okay, I got one full form. What about others? Yeah, please do write. Check your spellings. Yes, you can write for all of them. It's not like that you have to write one of them. If you know, you can write for full forms for all of them. Also, solve this question as well. Okay, so we'll move forward to the next one. So the answer for this question is all of them are involved in carbohydrate metabolism. So this EMP, HMP, ED, all of them are characteristics process of this carbohydrate metabolism cycle. Okay, so we'll move forward to the next one. Question number 16 is it's a true or false based question, so it's pretty simple. So anaerobic utilization of glucose happens in fermentation. True or false? This is kind of similar to the past two questions that we have seen, which was talking about glucose ATP conversion, as well as which one? Which question? This one. Um, it occurs only in aerobes. So please keep that in mind and solve this question. It's pretty simple. I have given you all the hints. Or what hint? I have given you answer, just in different words. So the answer for this question is true. That, as we have seen in the past question also, this glucose fermentation, glucose utilization happens in both the process, aerobic as well as fermentative. So that's why this answer for this question is true. You have seen right in the past question, question number 14, it occurs only in aerobes. So no. It can be used by the anaerobic bacteria as well in the process of fermentation. So that is why the answer for this particular question, question number 16, is true. Moving forward to the next one. So this is the glycolysis pathway. As we can see, we have a molecule of glucose, and we know that here you can see, right? I'll change the color of my pen. Yeah, this ATP is converted to ADP. So here, what is happening? The process, the glucose, the ATP is being utilized by the glucose. Okay, so here the energy is being utilized. Okay, so the first process, always there is an utilization of energy, but the net production of energy is more. Okay, so when the net production of energy is more, so we can say that glycolysis pathway is energy generating pathway. So in the first step, hexokinase is employed for the conversion of glucose to glucose 6 phosphate. Further, the next, when we say ACE, anything is ASE. So all of these are the catalyst. All of these are the catalyst. Okay. So when we whatever ends with ase is always the catalyst. So in the first process, glucose is by hexokinase is used. It catalyzes this reaction. So glucose is converted to glucose 6 phosphate. Further, phosphoglucose isomerase is used and the glucose 6 phosphate is converted to fructose. So what is the change between glucose and fructose? You can see in this part. Okay, here we can see the difference between glucose and fructose. Here also ATP is converted to ADP, means energy is utilized, not produced. Further, here the third enzyme, phosphofructokinase, comes under picture, which converts fructose 6 phosphate to fructose 1,6 biphosphate. Here, different products are produced. Either directly aldose will be used and dihydroxyacetone phosphate will be produced, or it can be directly produced glyceraldehyde 3 phosphate. The compound that has been produced by the aldase catalysis goes to trioses phosphate isomerase. It is utilized, it is a catalyst, and it forms the similar product, that is glyceraldehyde 3 phosphate. Here we can see that the energy is producing. Okay. So further, all other processes can be seen. And finally, we see that ATP, adenosine triphosphate, is being converted from adenosine diphosphate. So energy is generated and we get pyruvate at the end. So this is glycolysis pathway. This is the example of Krebs cycle, which is also known as citric acid cycle. You are very well aware of this cycle. Okay, citrate is there, which is converted to cis acetonitrate, then isocitrate, then alpha ketoglucarate, succinyl CoA. Okay, co-metabolic enzymes and everything, succinate, fumarate, malate, and oxaloacetate. So these are the process which comes after pyruvate is formed. Okay, here pyruvate is forming in the glycolysis pathway. From here, it is being converted to other forms. Okay, it's starting with pyruvate. Okay, so please take the picture of this, note it down so that we can move forward to the next one. Okay, so the third part is electron transport chain. Okay, so here NADH is converted to NAD plus, in oxidized, it is oxidized. So we can see the yellow one is oxidized form, red one is reduced form, and this slashed is electron transport. So how electron is transported from one part to the another. So you can take the picture of this so that we can move forward. And I would like to say also, electron transport chain is different from eukaryotic to prokaryotic. Okay. So the question number 17 is, which of the following is present in the archaeal membrane? We are talking about archaeal membrane. You remember like three domain system, archaea, eukaryotes and prokaryotes. So which of the following is present in archaeal membrane? D-glycerol, L-glycerol, ester linkage or ether linkage? So the answer for this question is L-glycerol ether linkages. Okay, please note it down. We'll move forward to the next one. So the next question is, sulfate ion can be used as an electron acceptor in anaerobic respiration in some facultative bacteria. We have seen this such kind of questions previously. What can be the result? True or false? So the answer is true. Okay. So this can be used as electron receptor in anaerobic respiration and some facultative bacteria. We have seen like fermentation, sulfate, nitrate, that question, remember? So oxygen photosynthesis requires carbon dioxide. Is it true or false? We'll see the solution now. It's true. Oxygen photosynthesis requires carbon dioxide. Okay. So the next question number 19 is, energy tapped in during chemoheterotrophy from inorganic molecules is more than that of glucose. This is pretty much simple. You know the answer. So true or false? This has been discussed multiple times in the lecture. We are comparing glucose, which is an organic molecule, to an inorganic molecule that can be sulfate, nitrate, and everything. You can see in your environment also which kind of bacteria grows more, aerobic or anaerobic. Okay, it all depends upon the energy. Microbial diversity depends upon energy. More energy, more microbes. So the answer is obviously false. More energy is produced from the organic molecules. That's why we see more aerobic bacteria. Coming to the next question, metabolic similarity frequently maps well with the functional similarity in microbes. Is it true or false? Solution is true. Metabolic similarity frequently maps well with the functional similarity. If they have same function, they will surely have the same metabolism. So this has been proven multiple times. If they have some kind of same metabolism that if all of them can metabolize a particular compound, they will perform a similar kind of functions. So which of the following is not found in a hot spring? This question is for metabolic diversity. Which of the following is not found in hot spring? So you all well know that why hot spring is hot. It's because of the presence of sulfur. Okay. So in this option, just to confuse you, there are different type of sulfur and non-sulfur bacteria. So which of the following is not found in hot spring? So the answer to this question is green sulfur bacteria. Okay, they are not found in the hot spring. Even though we call them sulfur bacteria, they're not found in the hot spring. The next question is: a genetic marker used for building phylogenetic trees should be universally present in all the bacteria. Is it true or false? This is pretty simple. If you want to make a tree, we need a root, right? So with that, you can solve this question. So the answer to this question is true. Okay, they will always have a common root. Then only this phylogenetic tree can be constructed. The next question is: oxidative phosphorylation happens in cellular respiration. Is it true or false? So the answer to this question is true. Oxidative phosphorylation happens in cellular respiration. The next question is: glucose 1p is being converted to glucose 6p. KQ is 17. Calculate delta G in calories per mole at 38 degrees Celsius 1 atm. Concentration of glucose is 1 raised to power 10 raised to, 1 into 10 raised to power minus 4 molar, and glucose 1p is 3 into 10 raised to power minus 5 molar. This is numerical based question. Please solve this. I will show you in the next slides. I will show you all the steps to solve this. Just to match it with yours, you can solve this question. So solution for this question is to calculate the Gibbs free energy change glucose 1p to glucose 6p. Okay, this is our reaction. So this is the equation delta G is equal to delta G naught plus RT ln Q. What is delta G naught? Delta G naught is minus RT ln Q. This is calculated, this is standard Gibbs free energy change. So R is 1.987 calories per mole per Kelvin. This is universal gas constant. T is 38 degrees Celsius. So we need to convert it into Kelvin because you can see that R is given in Kelvin. So we got 311 K. KAQ is 17. It is given in the question. And what is Q? Glucose 6P divided by glucose 1P. So this was the equation by product divided by reactants. No, right? This is how we calculate Q. So after doing this, we need to follow different steps to calculate the delta G. So delta G naught is what? 1.987 RTLNQ, right? So 1.987 into 311 into ln7. So ln7 is what? 2.833. This you can calculate by using a calculator. So delta G naught, what we got is minus 1748.7 calories per mole. Now we need to calculate the Q, right? So Q is this product minus reactant, which came out to be as 3.33. So ln Q is what? 1.203. Now next step is to calculate delta G. So this is used for calculation of delta G. It was what? Standard Gibbs free energy plus RT ln Q. Q means product divided by reactants. So simply we got delta G. So this negative G value indicates that the conversion of glucose from 1P to 6P is thermodynamically favorable under the given conditions. When the G value is negative, the reaction is thermodynamically favorable. If it is not, then it cannot be favored by thermodynamics. Please note down the steps. We'll move forward to the next question. The next question is energy yield. So there are two questions. So these questions are kind of open-ended questions. There can be multiple solutions for a particular questions. Okay. So there will be a particular term which can be called in a multiple ways. Okay. I hope you got this. So the thing is, there are now from question number 25 to maybe question number 30, there will be multiple questions, open-ended questions. So these are two questions, 25 and 26. Please write down the answer to these questions in the comments and we will see how many other solutions that we can have other than you guys are showing. So the solution for these questions are, for question number 25, it is inorganic compound. Even though if you write sulfate or nitrate that the other people have answered, that is also correct. As I have told, both of them are inorganic compounds. So you'll get full marks for this question. What about question number 26? Anamorphous? Yes, it can be used for removing nitrate. Other? What can be the other solution? Think. Yes. First can be full form anaerobic ammonium oxidation, which is nitrate removal process. The next set of questions are these. Please write down the answer of all of them. So the answer of first question is phylogenetically. Phylogenetically similar microbes may be functionally dissimilar. Okay. Enteric bacteria are found in intestinal tract and can be harmful or useful. Okay. No issue with that. Dash attaches to the surface of the prey and acquire nutrients from cytoplasm or periplasm. So either it can be epibiotic predators and parasites are epibiotic predators. You know that. Note this down so that we can move forward to the next one. The next question is Dash is not always purple and produce an array of carotenoids. Green sulfur bacteria form intimate two membrane associations with a Dash bacteria. Dash microbes is known to completely oxidize ammonia to nitrate. So the question answer for first question is purple non-sulfur bacteria or photoheterotrophs. Purple non-sulfur bacteria are also photoheterotrophs. The next question is the solution is chemoorganotropic or consortium. For the last one, it is ammonia oxidizers or nitrite oxidizers or nitrifiers or nitrosomnus and nitrobacter. Ammonia oxidizers are nitrosomnus and nitrite oxidizers are nitrobacter. And both of them combinedly are called nitrifiers. So that is all for today. So we'll see you all in the next session. Have a good night and have a good weekend. Not weekend, have a good week. Okay. Thank you, everyone. I'll upload all these lectures. Okay, so they won't